Children's Hospital in Boston. Kunkel is a renowned geneticist in muscular dystrophy. He's also the co-director of the Wellstone at the University of Massachusetts. And Lou is going to talk on modeling FSHD and zebrafish with therapy development. Thank you, Dr. Kunkel.
Thank you, Dan, and I thank the FSH Society for inviting me to present to you. I was asked to tell a little bit before I started about the Wellstone Center, but Peter has actually done some of that. I'm co-director with Charlie Emerson of a Paul Wellstone Center grant at the University of Massachusetts, Worcester. And our project is basically to understand, first, the genetic modifiers of FSHD, understand how one could develop therapies as another project, and then create animal models on which to test those therapies that you're trying to develop. And so I'm going to tell you just one portion of what's happening at our center, and that's related to our work with trying to model DUX4 misexpression in zebrafish. And I'm going to tell you a little bit about zebrafish before I start. All right. So why would we want to use zebrafish? Well, we can generate huge numbers of animals very quickly at very, very low cost as opposed to mice. These animals, these fish, develop within 24 hours, and they're out of their egg sac, and they're starting to swim within 2 days post-fertilization. So development occurs extremely rapidly. Their muscular dystrophy phenotype, which I'm going to show you in a few moments, is easily detectable by an abnormality of the way polarized light shines through these transparent fish. And one of the best things about them is they're a live organism which is permeable to small molecules. So if you had a therapeutic drug, you just put it in their water, and you can ask for it to correct for the phenotype of that animal. And I'm going to show you examples of that. So what I'd like to do today is tell you about how our final goal is to do drug screening in zebrafish models of FSHD. And I'm going to first show you not about FSHD, but I'm going to show you an example utilizing these models for Duchenne dystrophy, for dystrophin deficiency, and how successful that's been. And then I'm going to tell you about what misexpression of DUX4 does in an embryo, a developing fish embryo. And then I'm going to try to establish a stable line producing DUX4 so that we can start to try to characterize and do drug therapy screening in a live organism. So first, I'm going to tell you about chemical screens in dystrophin-deficient zebrafish to look for a functional modifier. So the way this is done is you take the fish, you mate them, and the SAPG is a dystrophin-deficient fish. It's an autosomal recessive disorder. So 25% of the animals are affected, the remainder are unaffected. We then take chemical libraries that are available to us at Harvard as well as at other institutions. These are libraries of known mode of action compounds. So these are drugs that you take, some of you take, for various different things. They include aspirin, they include Viagra, they include all sorts of different drugs that are all available for human use, already approved for human use, many of them. And what we're going to do is we're going to put them in mixtures in little microtiter wells with 20 fish in each well and ask, can we see for correction of the phenotype? And we use this Birefringence assay as our assay, and that's very characteristic. There's what the polarized light does in normal muscle at a 4-day-old zebrafish. That's 4 days post-fertilization. And this is what happens to that fish when they don't have dystrophin. You can appreciate the very, very abnormal structure of the muscle. It's going in all different directions. Very severe. They almost all die. So we're now going to take these fish and treat them. Here's 2 clutches of 20 fish each, and this is what happens. If you take one clutch, you can see 5 of those fish, which is the predicted 25%, are affected still, even though they were treated with drug. Yet only 1 fish out of the remainder in the second set, shown on the other side, are showing this abnormal birefringence. That's a drug that's effective in preventing the onset of this disorder, because this drug is given from fertilization. We end up with a whole series of drugs that actually have an effect. 6 of these compounds in this list all impact the same pathway. They say that pathway is very important in trying to correct for dystrophin deficiency. So we've learned a pathway. So now what we're going to try to do is take the same drugs and we're going to treat fish after they show symptoms of the disease. Can those same drugs work in that assay? And so this is what we call the 4-day assay. So we start 4 days, we pick the fish that are affected by the birefringence, and we treat them for an additional 30 days. And we do it in triplicate. You'll appreciate on the bottom line are drugs that are toxic long term. The blue line, I believe it's the blue line, is untreated fish. Most of them die. Some do live out. What I want you to look at is the red line, which is between wild-type fish, that means dystrophin normal, and treated fish. That particular drug is a PDE5 inhibitor, and it has actually been shown by others in mice to work. PDE5 inhibitors seem to work in mouse models of muscular dystrophy, thus verifying our fish screen. So I just wanted to give you an example of the fish screening, and now go back to FSHD and ask first, what does misexpression of DUX4, what does the phenotype look like? Now, if you remember, DUX4 is a homeobox transcription factor. Maybe you didn't remember that, but I remember that. and DUX4 is related to a number of other homeobox genes, including PAX3 and PAX7, which are expressed in skeletal muscle. And so what we've done with the help of the Wellstone Center is develop an expression plasmid that makes RNA for DUX4, which we can inject, as you see, into an egg, a fertilized egg, of a zebrafish embryo. And we're going to inject 10, 0.5, 0.2, 0.1 micrograms of RNA. And it turns out that, like others have shown in cell culture, in Xenopus, and in other systems, overexpression of DUX4 is extremely toxic. The vast majority, virtually all fish die when you add 10 micrograms, 10 picograms of RNA to the developing embryo. If you cut back to the levels where the DUX4 expression is thought to be at about 1 in 1,000 cells in patient samples, and you do that so that you put in just enough so that the RNA is diluted out by the time it hits somatogenesis in development to be about 1 in 1,000 cells. And that's about 0.2 picograms of RNA. This is 10 to the 5 molecules of RNA. That's molecules in that embryo. Many fish die at the higher levels. At the 0.2 level, you can see still some die, but in yellow, some survive, and they have an abnormal phenotype. And the phenotype is really quite striking. I'm going to show you. Let's go. Low levels of DUX4 resulted in asymmetric expression and abnormalities of the eyes, ears, and fins of these zebrafish embryos that were injected with 10 to the 5 molecules. And you can see that pictured here. There's an eye on one side and not on the other. There's a fin on one side, not on the other. This is actually quantifiable, and you can show that this is a phenotype that one sees as a result of DUX4 expression in these fish. In addition, these fish also have abnormal birefringence of their skeletal muscle, as you can see by the yellow arrows pointing to places where the muscle is disorganized. So at very, very, very low levels, you can recapitulate a lot of what one sees in the symptoms of FSHD in patients, in zebrafish. So conclusions, very small amounts can cause abnormalities of the face, eyes, and fin muscles. DUX4 is perturbed myogenesis in the face and eyes, and zebrafish can start to serve as a model for muscular dystrophy, for FSHD. I'd like to briefly, if I can, Dan, try to establish a DUX4 transgenic line. So we're going to try to express now transgenically. That means I'm going to be able to control the expression. I'm not going to inject RNA. I'm going to control the expression to make a stable line on which I can do drug screens for therapy. And this utilizes the tamoxifen system, where you're actually going to remove part of the DNA and open up a red marker that can be attached to the DUX4 locus and show that you can actually turn that gene on during development. We have a number of constructs that we've made at this place, and you can see that when we do this, we can transgenically express, just like in the human muscle cells cultures, one can get very, very low levels of DUX4 expression in the skeletal muscle, as you can see in the middle panel and the left panel, where you can see the abnormal skeletal muscle. We're now going to set up an activity screen so that we're going to compare a normal fish, can't tell which direction it is, but where one sees no M. cherry, that is the red strain, whereas the fish that has the M. cherry, and we're going to monitor their activity in an activity chamber, which is quantifiable. And we can show that the normal non-DUX4 expressing fish are much more active in that assay than are the DUX4 fish. That's the expressing fish. That gives us an assay on which to now start our drug screens. So both the injection and the transgenic model of DUX4 give us a muscular dystrophy phenotype. Despite the primate-specific origins of DUX4, our zebrafish model is able to mimic some of the FSH patient phenotypes. This suggests that misexpression of DUX4 results in a toxic disease pathway that is conserved across vertebrates. We can use these fish to find these conserved elements and targets of DUX4 in development, and our future work will be to use this transgenic animal to do drug screening. And I'd like to acknowledge in the last slide Angela Litt, who is in my lab currently working on this project, as well as Hiro and Fedek, who are also part of the Wellstone Center, and acknowledge the FSH Society for supporting some of this work, as well as NICHD and the Wellstone Center. And I thank you.